Is this thing recording? I guess it doesn't matter, I suppose. All right, so this is Chapter 8, Part 1. This is a pretty important chapter. Some of it you guys will have covered before, almost certainly, and some of it you will have not covered before, almost certainly. So, and you can see I divided it into two parts, and I think that's because it's long, yeah? And there's a ton of information. So, Chapter 8, Part 1, that's what we're starting with now. And that we're going to talk about DNA structure, DNA replication, RNA structure, and then gene expression. And then Part 2, we'll talk about some other stuff, yeah? So, why is DNA so important? Does anyone feel like answering this? Maybe this is something that we could, I could take a little, see, is this thing recording? Yeah, it looks like it is. Okay. So how about it? Why is DNA so important? Neda? So therefore, it makes us. Anybody have anything else? What do you think? Pretty good? Very good, I would say, yeah. Certainly very much in line with what I intend to teach, yeah? Is there anything anybody's got to add to this? Were you thinking something a little different? How organisms will what? Behave. How are capable of behaving. What do you think about that? Very good. Yeah, absolutely true. Yeah, right. The more we know, right, what we know about the human genome can tell us, can make predictions about potential genetic health problems that people might have in the future, yeah? And because of that's actually, you know, part of the excitement and to some degree part of the peril of DNA technology, yeah? The more we learn about DNA, the more into somebody's personal genetic business the world can snoop, yeah? Which has its good parts and its bad parts. It's bad because, again, we have personal rights to privacy, but it's good to know in advance if you're going to have a genetic predisposition to heart disease, yeah? Or diabetes. If you're more genetically predisposed to diabetes, you can watch your sugar content. If you're more genetically predisposed to heart disease, well, you can take a medicine prophylactically or whatever. If you're more genetically susceptible to breast cancer, you can do something about that, yeah? I guess, what's her name, that famous actress lady? Yeah. How many people have heard, you know, it's really important news that you guys get from the tabloids, yeah? Where Angelina Jolie, I'm sitting here trashing her, but she actually did something to set an example, and she did something for her own personal health. Yeah, she got prophylactic mastectomies, double mastectomies, right, in order to reduce her family risk of breast cancer, right? And she made that decision based on genetic testing, right? She can have her DNA looked at, and she can see genes that make somebody much more susceptible for much higher risk breast cancer, yeah? Right? So those are fascinating, and to the extent that they're about biotechnology, and to the extent that microbiology class discusses microbiology or biotechnology, which it does in Chapter 9, this is relevant. But we're certainly here to talk more about microbes than we are to talk about Angelina Jolie. But what an actress, really. Can we just talk about it just for a minute? Just kidding. All right. What about this, right? Again, we can see genetic tests that tell us how microbes are going to behave. Did we see how that was relevant in some of our homework assignments? We absolutely did, right? KPC may not have been able to be stopped by Uncle Sam's Hospital, but Uncle Sam's Hospital could follow it very closely and see how the disease was transmitted from patient to patient, right? By looking at the DNA, right? Do you guys remember that part of that video that we saw? The guys in the technology center next door to the hospital were taking samples from people and looking at the DNA, and if they found the gene for KPC, they knew that person had microbes that had the KPC gene in them, yeah? Right? And that tells us not only what antibiotics to not bother using with those people, it also tells us we learn something about how disease is transmitted by following with DNA. Does that make sense? So DNA is pretty darn important, and it will become more and more directly relevant to the nursing profession as the years go by. Yeah? 10 years from now, it is very conceivable that along with looking at a patient's chart of their vital signs, right, and their medical history, you'll look at their genetic history and the results of genetic tests about microbes that we're trying to diagnose, as well as the diseases that they might have, yeah? All right, so DNA is important, and I've justified, hopefully, the reason that we learn about it today, yeah? Are you guys convinced? Are you guys now gospel converts to the need for learning about DNA? Okay. All right, so DNA is the code to make a protein, yeah? And I would thank Netta and Michael for making that so clear, yeah? DNA is a code to make a protein. A gene is a sequence of DNA which makes a single product, usually a protein. I think I had this in something of a, I'm going to put a little asterisk up here. I hope that doesn't mess anybody up. And I'll just put asterisk, usually. So it's usually a code to make a protein. DNA does not always, relevant DNA information is not always for making proteins. There are a few other things that we'll see, right? You don't need to get a headache about this now, but we will see in the future how DNA can be the code to make some other products that are relevant. All right, a chromosome is a long strand of DNA containing many genes. How many chromosomes, how many pieces of DNA do you have in the human genome? 23 kinds, 46 total, because we have doubles on every one because we're diploid organisms, yeah? Right? How many does most bacteria have? One piece of DNA, yeah? Good, good. All right. And then a genome, right? All 46 chromosomes plus the DNA in our mitochondria, that constitutes the human genome, yeah? But again, as we've learned, each of us has our own personal genome because no two people have the exact same DNA, yeah? And so a genome, we could say, is the genome of all the genes in a species like humans, an individual like you, but then again, if you slough off your DNA on your victim's neck at the crime scene, sorry, you guys wouldn't do that, yeah? That was me. That was me. No. No, it wasn't. It wasn't. Just kidding. Just kidding. Sorry. Anyway, so, right? Any cell in your body has the same genome, yeah? Your cells are genetically identical. So a genome, the term genome, can describe all the DNA of a species, all the DNA of an individual, or all the DNA of one particular cell, yeah? Does that make sense? That term is flexible based on the context. Okay. All right, so proteins are incredibly diverse at the molecular level, right? And as we've shown, right, this is the same slide you've seen probably once or twice already, yeah? You guys recognize all these? Basically, I use this slide to show you pictures of fancy, complicated proteins, yeah? Right? And you remember the primary, secondary, and tertiary structure, yada, 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 yeah? And again, we're made out of protein, as Neda pointed out, yeah? So fish scales, bird feathers, and of course, platypus fur. How many people knew that there was such a creature called a platypus? Duck-billed platypus. I remember one of my first semesters teaching this class, when I made this slide, I had a couple students say, you know, quit throwing internet hoaxes up in your slides. And they had never seen this creature, and they thought I was posting some internet hoax. Yeah? This is not an internet hoax. This is a real creature, a duck-billed platypus. It's adorable. It's got fuzzy fur. It's got webbed feet, and it's got a little duck bill. It's a real creature. Google it. Raise your hand if you've never heard of this creature and you're seeing it for the first time today. It is my great privilege to show you, behold, the duck-billed platypus. It's actually kind of an interesting little creature, and I'm so glad that I get, right? So if you learn one thing all day, it should probably be DNA. But if you learn two things, the other thing should be this, yeah? This is certainly more fun. Okay. Moving on. So DNA is important. DNA makes proteins. You're made out of proteins, yeah? What happens when your DNA gets messed up? Well, when DNA gets messed up, we get a lesson on how important it really is, yeah? When DNA gets messed up, here it's supposed to be GAG. It goes to GTG. We change one letter in DNA, and suddenly a normal protein that has this shape when it functions normally now has this shape, yeah? Do you think this protein will function as well as it did before? Probably not, yeah? So, change the DNA, you change the shape of the protein. You change the shape of the protein, you change the shape of the cell that is filled with that protein, yeah? And you go from a regular red blood cell to a sickle-shaped red blood cell, yeah? And if you mess up the protein and mess up the cell, then that cell doesn't flow through your blood vessels very easily. It can get stuck there, yeah? So mess up the DNA, mess up the protein, mess up the cell, mess up the circulation of the person, yeah? That's where sickle disease comes from, yeah? There are other weird complications that, which is that, that you mess up the cells in this particular way, and those cells, strangely enough, are resistant to the disease malaria, yeah? And you can see a correlation between where malaria is found in the Middle East and in India and Africa, and where you find people who have that disease, yeah? So that's more of a story of evolution than it is something else, but it's kind of an interesting story. Okay, so DNA is important. We see its importance when it works right. We see its importance when it goes wrong. All right, so DNA contains all the genes needed to make another cell, yeah? Any cell in your body has to have a copy of the genome in order to make all the proteins that cell depends on, yeah? This is an inaccurate map of the E. coli genome, yeah? So it's just one chromosome. We can, and it's circular. You guys remember that E. coli prokaryotic chromosome is circular, right? So we're drawing it as this circle, right? I happen to know that the size of the E. coli genome is about 3 million base pairs of DNA. Looks like it's 3.4, yeah? So, BP, what does that stand for? Base pair. You guys understand what a base pair is? If you don't, we'll talk about it in just a second. Yeah? KBP, what's that? What's the K stand for? Kilo, which is 1,000. So a kilobase pair. And how many kilobases do we have? 1,100 kilobases, yeah? This is 1.16 million base pairs, yeah? 1,000,000 is a million. You guys agree? Right? And if I know that it's 3 million base pairs to make an E. coli genome, then that should be, we should start here, and we should be at 3 million here, right? And it's showing 3 million here. That's wrong. It should be up here, yeah? Does that make sense? So this is slightly inaccurate. But we can see on here are all the genes we might expect are necessary to build a cell, yeah? We need to be able to make amino acids. We need to be able to make a membrane. We need to be able to copy DNA. We need to be able to make lipids, yeah? And so all those genes have a place on the E. coli genome. Does that make sense? So this is, again, not something you need to learn, but it's something that helps you understand that DNA is, your genome is like a book of blueprints for making all the stuff the cell needs to make, right? If the cell needs to divide, well, we're going to need a phospholipid bilayer in that new cell. We better go to membrane synthesis and lipid metabolism to make those phospholipids. Does that make sense? Right? If we need to build a protein, well, we better go to amino acid metabolism to make sure we have enough amino acids. Yeah? Does that make sense? Right? So all the stuff that E. coli needs to do to be alive, the plans for the machinery that does that stuff is right here. Okay. So, central dogma of molecular biology. Do you guys know what the term dogma means? Dogma usually pertains to something like religious belief. And this was, have you heard of Francis Crick? Of Watson and Crick fame, except his name is not spelled with a K. Why did I write a K there? I-C-K. Watson and Watson and Crick were the two guys who discovered the structure of DNA. And Crick said, well, we can expect, once he found the structure of DNA, it was very predictable what the basic layout of how DNA was going to work. And he made that prediction, and he said, well, the dogma is that DNA flows to RNA, and then that is used to make proteins, yeah? But the term dogma, if it implies religious belief, right, accepted without proof, when he said this, there was no proof. Since then, there has been extensive experimentation and proof that this actually happens, yeah? So what you're going to learn is not stuff that is theorized, it's stuff that's demonstrated, yeah? Is that good? All right, so we've got three main things, two of which we're going to talk about in Chapter 8, Part 1. The other we'll talk about later on. Replication we'll talk about first. Do you understand what that means? Replicate means to copy, right? Take a strand of DNA and make an exact copy of it. And again, that's necessary if you want to be able to reproduce, yeah? DNA replication is essential for cellular reproduction, yeah? Okay. Furthermore, DNA expression, gene expression, is essential for survival, right? In other words, the reason we have DNA is because we need the proteins. So expression effectively means making proteins. Is that clear? So a good example of that is my genome and my cells, yeah? My genome has all the instructions for making the proteins of my hair, my skin, my muscles, etc., yeah? But if I look in the my muscle cells, they are not making hair protein. Does that make sense? You agree? My muscle cells are making muscle proteins, yeah? In other words, they're looking at chapter 5 of the human genome, whereas my scalp cells, they're not making muscle proteins, right? They're making hair proteins, yeah? Right? So, thankfully, at my age. So the fact that my scalp cells are a different piece of DNA, that is called expression, yeah? My scalp cells are expressing hair proteins, yeah? My muscle cells are expressing muscle proteins. Got it? Right? We'll learn about the mechanism of gene expression. It involves a couple of steps. Transcription and translation, we'll learn those today. Okay? Questions? Doing all right. Okay. So, DNA structure. You guys hopefully already know the basic structure of DNA. It's a double helix, yeah? And we can see the double helix, right? Can you see all the little red, yellow, blue, gray, and white spheres here? What are those? So each of these little spots is an atom, yeah? DNA is a molecule. Ultimately, any molecule is made out of atoms. And so each of these little atoms, right, comes together to make, can you see the double helix in here? Right? You can see it twisting. Yeah? So this is a molecule of DNA that happens to be highlighting every single atom. Yeah? Okay. And it looks like a twisted ladder. There's rungs going across the middle. We'll see those in a second. Yeah? DNA is made of 4 kinds of modular subunits called nucleotides. Right? A nucleotide is a little piece of DNA. Yeah? Each nucleotide itself is made out of parts. The parts are a 5-carbon sugar, that's shown in blue here. That's the sugar. A phosphate, that's where it says PO, with a bunch of O's around it. PO4 is a phosphate. And then a nitrogen-containing nucleoside base. That's in the orange here. And we can see there's 4 different kinds. They're not all the same. Got it? Okay, so the 4 kinds of DNA bases, there's 2 kinds that have double rings, yeah? This one and this one have 2 rings each, yeah? We call those purines, and adenine and guanine are 2 purines, yeah? Pyrimidines are these 2, thymine and cytosine, yeah? And thymine and cytosine are just 1 ring, yeah? And as it turns out, A always matches with T, C always matches with G, yeah? So TA, and we can see the T and the T from this side and the A from this side are held together with hydrogen bonds, yeah? 2 hydrogen bonds here, 3 hydrogen bonds holding C's to G's. Can you see that? All right, so now we're getting to the term base pair, yeah? If you didn't know what a base pair is, this is a base pair, yeah? These are DNA bases. This is our T thymine base, adenine base, and they always pair together, yeah? So basically, a base pair is a rung of DNA, and we can equate how long a piece of DNA is, and therefore how much information is in a piece of DNA by looking at the number of rungs of that ladder. Does that make sense? A ladder with 1,000 rungs is taller than a ladder with 100 rungs, and if each of those rungs has DNA information, then the longer strand has more information. Yeah, Michael? We'll talk about that in a second. That's RNA and not DNA. Okay, so let's see. Covalent bonds link all the parts of the bases on each of the sides, yeah? Covalent bonds hold phosphates to sugars to phosphates to sugars. Covalent bonds hold sugars to base, sugar to base, yeah? Hydrogen bonds link together the bases across the rung, holding the strands together, yeah? Which are stronger, covalent or hydrogen bonds? Covalent bonds are stronger, yeah? Hydrogen bonds are fairly weak, but again, this is built into the structure and it's important, right? Hydrogen bonds are weak, but each individual zip in a zipper is also weak, yeah? But the collection of all the zips along the length of the zipper is strong enough to hold your jacket together, yeah? Does that make sense? Right? Furthermore, the fact that you have a bunch of individual zips allows you to zip up and zip down your jacket very easily, so you can put it on and take it off really easily, yeah? So, and the way that zipper works, the little zipper head, is it pulls apart each little zip one by one, yeah? Right? Even if, right, they're weak, but all together they're strong, but just because they're strong all together doesn't mean that they're easily broken one by one by one by one. Does that make sense? And so that's going to be important to how DNA is expressed, that is, how we read the DNA to make the proteins, and also how we copy the DNA. Does that make sense? Okay. So, one other thing that's important is we have this fancy word here about anti-parallel. Okay? You guys understand that term? Probably not. This might be a new term for you. So, but you do know the word parallel, yeah? So parallel, we might think of the opposite of parallel as something like perpendicular, yeah? But this is a different word. This is anti-parallel, yeah? So show me parallel with your hands, yeah? So I usually show, or try fingers. Give me index fingers, yeah? There's your parallel fingers. All right, so this is anti-parallel. Does that make sense? With one pointing upwards and one pointing down. So parallel involves no directionality, no up, no down, just lines, yeah? If we imagine that the lines have direction, right? We point with fingers, right? Then I'm pointing up with both of these, but DNA points up with one and down with the other. Now that I've mentioned that, can you see that here? Right? You can see that on the left side, we've got a phosphate on the top, we've got an OH group at the bottom. If we look at the right side, we see it's the opposite. We have an OH at the top and a phosphate at the bottom. See that? Yeah. Also, we can see that each of these sugars in purple here, these sugars have a little oxygen at their top, yeah? Each one with the oxygen pointing up. Here, each one of the oxygen is pointing downward, yeah? So, the two strands are looking at each other backwards, sort of, right? They're running parallel, but they're parallel in opposite directions. Okay, and again, the sequence of bases contains the information to make proteins, yeah? So, these strands bound together, right? This is our double helix of DNA, and we have our hydrogen bonds holding them together, but the amount of information correlates to the length of the strand of DNA, right? And that correlates to the number of base pairs we have. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 base pairs here, 11 little bits of DNA information. Does that make sense? Okay. So, now DNA replication. In other words, copying DNA. Why do we need to copy DNA? What's the point of copying DNA? To make new cells, yeah? Without copying DNA, right? If we don't have the blueprints, we can't make the proteins. If we can't make the proteins, we can't live. So if we reproduce, but one of them doesn't have the DNA, it's not going to be able to live, yeah? So in order to reproduce, we have to have a copy of the DNA so that that new cell is viable. so that new cell can live. Does that make sense? You guys got that? Okay. And that goes for everything, right? Whether it's us or a prokaryotic creature, yeah? Every one of our cells has to have, instead of one piece of DNA copied, has to have 46 pieces of DNA copied in roughly the same basic way, yeah? Is that clear? Right? So, DNA is DNA from one creature to another. We just have more pieces of DNA, yeah? And so having those extra pieces of DNA, we still need all 46 pieces in order to have a viable cell. How many cells is your body made out of? Trillions, yeah? And so every cell that was formed had to have all the DNA copied for it before it began its cellular life. Does that make sense? Okay. So, again, looks like this slide is a bit redundant. Hydrogen bonding between the DNA bases, we know about that. A always matches with T. C always matches with G. Yeah? Okay. So, here is the basics of how that plays out when DNA needs to be copied. Once it was known by Watson and Crick, the shape and structure of DNA, right, to some degree, that told us how DNA was copied, yeah? And here, it's even more evident, yeah? This is the very simplest diagram of how it works, but in here is the biggest chunk of important information as to how DNA is copied, right? And it's easy, yeah? Pull apart the strands and then match up each strand with little nucleotides, little pieces of DNA. Guys with me? Right? So, you guys could do this, yeah? I cover up one side. Can you still see this side? A always matches with T. C always matches with G always matches with A with T with, right? And if I did the opposite, T matches with G with C with T with A with, right? After a while, so at some point, right, once you know the pattern, you know that A matches with T and can't match with anything else, you know that C matches with G and can't match with anything else, then it becomes a literal no-brainer, yeah? You don't need to think in order to do this. You just need to remember. You just need to follow the protocol. Does that make sense? Right? So, this is, instead of you thinking about it, this is a job that could be replaced by a robot, right? In other words, cellular machinery does this, and cellular machinery doesn't think about it. It just does it mechanically. Does that make sense? So we pull it apart, and we match up the bases, and then we have two new strands, yeah? And so theoretically, right, this shows 1, 2, 3, 4, 5 bases being copied, but again, for a E. coli genome, it's not 5 bases, it's 3 million, yeah? And for a human genome, it's not 3 million, it's 6 billion bases, yeah? And this mechanism can copy all 6 billion bases of your DNA, theoretically, with 0 mistakes, yeah? What do you think? Pretty remarkable, yeah? You think about your chances of getting a book and copying it word for word, and how many mistakes you're going to make trying to stay awake doing that, yeah? I would certainly make many mistakes. But again, you have trillions of cells. That means that DNA replication has happened in your body millions or trillions of times, yeah? Right? And again, virtually, not 0 mistakes, but extremely few, right? So few that it's virtually no mistakes, right? Does that make sense? Incredibly, right? And at least theoretically, it's possible to do that. If there's only A with T, only C with G, then in theory, you could do it with no mistakes, and in practice, living things come very, very close to that perfection. Questions? Yeah.
Did you notice that it looks like instead of equal time, middle age about C?
You know, I've been given this lecture. I've used this slide for almost 10 years now, and nobody has ever mentioned that. And I've blathered on about 0 mistakes, yeah? And it looks like the guy making the figure didn't even notice, yeah? And nobody proofread him. DNA, one of the reasons it's so accurate is because the enzymes that get this done do something called proofreading. DNA enzymes that do this job check their work, yeah? But I have been blathering on year after year, semester after semester, and I've never checked to make sure that this figure didn't have any typos in it. And obviously it does, right? There's supposed to be a G going here first before this A matches with this T, yeah? Whoopsie. Thanks a lot. Now I got to fix my PowerPoint. See the reward you get for helping, yeah? That's the way. Ain't that the way the world works, yeah? Yeah. So kidding aside, dang it, now I really will have to fix this. Although conceivably I could go another 10 years before anybody else noticed, yeah? Whoa, all right. Anyway, you guys get the idea? In theory, this could be done with 0 mistakes, and this is not a real cell. This was made by a person, and as you can see, the person does make mistakes, yeah? All right, so DNA replication is semi-conservative, yeah? And we can see that both here and here, semi-conservative does not in any way reflect a political leaning of DNA slightly to the right. It actually means DNA, each new strand is half old DNA and half new DNA, right? So the old DNA is conserved. That is, it's the A's, G's, C's, and T's are all the old piece, yeah? And so each new strand is half old in blue and half new in gray. Guys got it? Likewise, in this picture, each new strand, right, we pull apart the DNA here, and you can see that a little more. This is a little more realism. It's still a cartoon. It's still a figure, but it's a little more accurate, yeah, because we see the double helical shape, and we can see that the two sides are not being assembled in exactly the same way. On both sides, old DNA is in purple and new DNA is in pink. You guys see that? All right. But is there a difference to the way the left side and the right side are being assembled? Yeah, so they're being assembled oppositely. Yeah? Do you guys see that? So let me ask, which one is being assembled from the bottom up and which strand is being assembled from the top down? Can you tell from the picture? See if you can visualize it. Yeah? As the single strand is separated into two strands, you can see here we have something called the replication fork. Yeah? As the DNA is separated, is the replication fork going to go upwards or downwards? Upwards. You guys see that? Right? So which strand is being copied upwards and which strand is being copied downwards? Can you see that? Raise your hand. Match them up, left and right, upwards and downwards. Luisa's got her hand raised. Neda's got her hand raised. David's is doing something to, right? If you're doing something physical to try to solve this, if you're making a guess in your mind, you're more likely to learn, yeah? All right, Luisa, let me hear it. Left is going downward, right is going up. What do you guys think?
I
It's the opposite, yeah. So Netta and Luisa got it wrong. It's going upwards here, yeah? It's made here. These are paired up. The next one going to be paired up is this guy, yeah? And then this guy, and onwards upward, yeah? You guys see that? And then here, these guys are paired, and the next one is this one, and then this one, and then this one, yeah? So this one is going downwards, yeah? So raise your hand if you can explain why this guy's going up and this guy's going down. That's because DNA is, complete the sentence, DNA is anti-parallel. High five to anybody who was thinking that, yeah? Does that make sense? Right? So what's implicit here is DNA is directional, and therefore DNA is assembled in a directional manner, yeah? And it only works in one way, right? Maybe think again of things like Legos, yeah? Legos are assembled bottom, right, with the bumps on the top to the holes on the bottom. Does that make sense? Legos, bumps on the top, holes on the bottom. Anyone not know what Legos are? Do you know what Legos are? You do, okay. When were you playing with Legos? What was your age when you came to this country?
No, I do like child play for part-time.
Ah, okay.
Play with kids.
All right. Play with kids. Sounds like you're not playing with kids, you're playing with the Legos. Sorry, I shouldn't tease. I'm just pulling your chain a little bit. Okay. So, sorry about that. I hope you don't mind. Okay, so you get the idea, yeah? So my next question is, which of these strands has the easier job? It's kind of a weird question, but put a guess out there and see if you can get it right. Which job is easier and which job is harder? The guess that they're the same is a legitimate guess, but I will make the argument that that guess is wrong, yeah? Assembling the left side is not the same as assembling the right side, and one job is easier than the other. See if you can think of a guess as to which job is easier, and think if you can guess of a rationale as to why it's easier. Amaryllis, do you have a guess? Which side is easier? Right side is easier? I would say the left side is easier, yeah? So if the left side's got the easier job, curses! You know what happens if you get it wrong? Nothing, right? Who cares? You learn something, yeah, exactly. You're more likely to learn when you put it out there and get it wrong, yeah? So why do I think that the left side is easier to make than the right side? Anyone give me a rationale for why the left side is easier to assemble than the right side? Stronger. What would make it stronger? When those two strands are finished, they should be chemically identical, yeah? One should be just as strong as the other, right? So, but that's when we're done, right? In the process of building, why is the left side easier to build than the right side? Netta? Replication fork, yeah? As the replication fork goes up, do you guys hear what Netta's saying? I think she's got it, yeah? Belt it out loud, please.
As the replication fork goes upward, and this left side goes upward too, so it's easier to build upward.
Does that make sense? You guys hear that? Say it again, please.
As the replication fork goes upward, open the strands of DNA, the left strand is easier to build upward.
It's going up too, and it can follow right behind the replication fork. What about the right side? It's going down, yeah? So, where is it going to start? Well, maybe it started here. What about making this part? How are we going to make this? Yeah? You guys understand? Five prime end to 3 prime end. Do you guys see that? That's something you won't those are technical terms that I'm not going to require you guys to know, but I'll explain that in a second, yeah? 5' and 3' have to do with the directionality of DNA, and I'll show that in a second. Okay? All right, so what Netta is saying is that this one can follow right behind the strand as it opens. And as soon as this C and G are separated, this guy is ready with another G to put on that C. Does that make sense? Whereas this one, right, if it wants to copy this section, it's going to have to start here. Yeah? Right? It can't start up here because this is still stuck together. Yeah? And so, but once it starts, it's going to go, it can only go down to here. Does that make sense? And then its job is done because this part already got done last time. Yeah? Does that make sense? And so in order to do this, we have to wait until the replication fork opens. And then after doing this section, we can do this section. But again, we started here, so we're only getting from here to here. Does that make sense? Guys with me there? So what essentially that means is that the leading strand is synthesized continuously, and the lagging strand is synthesized discontinuously. Do you guys see that? You guys, that makes sense to you?
Yeah.
Okay, good, good. All right, so let's go back. So let me see if I can answer David's question about 5' to 3'. It says it here. DNA polymerase is the enzyme that makes this happen, right? I show you pictures with free nucleotides magically coming to the surface of the template strand of DNA. Does that make sense? You guys understand what I mean by template strand? Right? The one we're copying off, yeah? That is the template, yeah? And so these things magically float the T to the A, the C to the G. Should that happen? Ultimately, yeah, right? T has hydrogen bonds that naturally match the A. C can make hydrogen bonds that naturally fit the G, yeah? So they would sort of naturally float together, but again, that's like saying, sure, the basketball naturally fits in the hoop, yeah? It does fit in the hoop, but if you're taking a half-court shot, it's going to take you a while before you hit 3 in a row. Doesn't that make sense? Right? Likewise, any chemical reaction, and the assembly of a piece of DNA is a chemical reaction. Chemical reactions in living things happen with the help of what? Enzymes, yeah? And so there's an enzyme, DNA polymerase, that is helping the synthesis of DNA, yeah? And we can see that here. DNA polymerase is the enzyme that you need to lay down the track. And there are lots of other enzymes that you will not need to know, at least for this chapter. We'll talk about a couple on in chapter 9. But for now, the only enzyme you need to know is the enzyme that lays down the track. DNA polymerase basically is the guy that grabs the nucleotides and matches them up with the template strand. Yeah? And here we can see, is this the leading strand or the lagging strand? On the top here. Leading or lagging? Anybody but Netta. Netta can't say it because I know she understands the concept. Yeah? Which one's the leading and which one's the lagging? Top and bottom, leading versus lagging, which one is which? Ah, hands are going up. Tori. Top is leading or lagging? Leading. Yeah? How is it leading? Well, again, we have some other enzymes. You don't need to know all these enzymes, but this one pulls apart the DNA. As soon as the DNA is pulled apart, this guy's ready to lay down track, yeah? Does that make sense? What about this guy, right? Here is the same enzyme, DNA polymerase, right? And it's making track going in the opposite direction of the movement of the replication fork. And here it's going, and it's going to bump into this one that's already been made. And in order to make this strand, it's going to have to let go and then start here. And then it's only going to be able to do this part. And if it wants to do this part, it has to wait for the replication fork to open, right? So this guy never has to let go, yeah? He can just ride the rail, so to speak, and he can follow it along until the strand is done. This guy has to build, let go, build, let go, build, let go, build, let go. Does that make sense? So one strand is made continuously, the other is made discontinuously, and made piece by piece by piece by piece. And the pieces, these pieces here and here and here and here are called Okazaki fragments, yeah? And why are they called Okazaki fragments? Because they were discovered by a husband and wife research team named Smith. No, kidding. They were named Okazaki. Yeah, that was their last name. Does that make sense? Right? And the reason that one is built continuously and the other is built discontinuously is because the enzyme only works in one direction. Yeah? And DNA has an up direction and a down direction because it's anti-parallel. Yeah? All right. So that direction is technically called the 5' to 3' direction. And so David asked, what's up with that? So I'll explain briefly. And this is something that nobody needs to know, but I'm just sort of explaining for a second. Yeah? All right. So this is a ribose sugar. Yeah? This is a 5-carbon sugar. And sugar chemists spend a lot of time thinking about sugar, so they number the carbons that the sugar is made out of. This is carbon number 1. This is carbon number 2. This is carbon number 3, and it has the OH coming off of it. So off of the OH, this is the 3' carbon. Yeah? This is the 4' carbon, and this one, the 5' carbon, has a phosphate coming off of it, PO4. Yeah? So this phosphate attaches 5' to 3'. 5' attaches to 3'. I don't know what this arrow is doing here. That arrow is sort of a typo, I would say, or going after explaining a different concept. This phosphate is binding to this hydroxy, 5' phosphate to 3' hydroxy. Does that make sense? And again, if you don't remember that, well, that may be a problem in another class, but not this class, yeah? Right? You should know that DNA polymerase works in one direction only, yeah? And so because the DNA is anti-parallel, it's easy to make the leading strand. The lagging strand has to be made piece by piece by piece. The pieces are called Okazaki fragments. Those pieces, eventually, we'll learn, are linked together by ligase enzymes. So other ligase enzymes link together the pieces. There are primers that get the reaction going, and RNA primase lands on those to make those so that the DNA polymerase can get its job going, and so on. But again, those are all details that you don't need to know. Questions? There's a bunch of enzymes here. DNA gyrase, ligase, helicase, methylases. It's topoisomerases. The only one you need to know is that guy. Yeah, DNA polymerase. We'll highlight him. That guy, yeah? Good? All right. And what does DNA polymerase do? It lays down the track, right? It matches A's with T's. It matches C's with G's. And we can see how many DNA, if we have a circular piece of DNA, right? We have 2 replication forks, yeah? This is prokaryotic DNA replication. It always starts in about the same spot. There's a place called the origin of replication, and that's where DNA polymerase starts. Does that make sense? Right? And so where is DNA? If I look at this circle, you guys see this one? We start here, then we go here, then we go here. You see this as a continuous step-by-step process of copying one strand of DNA into 2. Yeah? All right. Can you see where DNA polymerase would be in this picture? Where do you think, right? We've got one strand being turned into 2 strands. where is DNA polymerase right now? If we could zoom in on this strand of DNA, I'll start here. Somebody scream when they think my index finger is touching a DNA polymerase. You should be screaming here, yeah? Ah! Okay. Oh, nicely done, see? Sends chills down your spine, doesn't it? Okay. Guys got it? Right? Here is where the DNA is being copied, and this is where DNA polymerase is doing its work, yeah? And so these guys, the replication forks will go this way and they'll go down, and as they continue, they're going further and further, and finally they get to the opposite end from the origin of replication. Does that make sense? And that's where the two strands separate. Okay. All right, so that's replication, yeah? Guys got it? Take a deep breath. We are done with replication, and I've been jabbering for almost an hour. Sorry about that. You guys tired? Do you need a break?